Okay, guys. Hey, how you doing? Mr. Mawson here. We're going to do Unit 7, which is Chapter 17, Lymphatic and Immune Systems, and Chapter 18, Respiratory. So this is going to be your 20-minute video lecture. This is going to be very quick, very fast, lots of highlights here, okay? So lymphatic and immune system starts on page 341, 341 in your book, Chapter 17. Again, starting out, we're going to go through slides quick. Um, so lymphatic, basically starting out, we're discussing just lymph as a general rule of thumb, right? You want to kind of remember that we're talking about, sorry, I'm making sure my time is going. Yeah, we're talking about a system that 15 or so percent of the fluid that gets pushed out into our tissues from our bloodstream, from our cardiovascular system, ends up needing to kind of get catched by something. And the catcher here is lymphatic system. Lymphatic system takes about 15% of that fluid that we lose for every capillary exchange and dumps it back into lymphatic system. When it does that, it does a few things. Page 342 has a fast fact and body of work that highlight the things that the lymphatic system does. But again, big things: maintenance of fluid, absorption of fats. But the big thing is immunity. Our lymph nodes and other lymphatic vessels hold on to our immune cells, let them mature, and give our immune cells a chance to see any pathogens, see any foreign objects that get pushed through the lymphatic system. Everything runs back through the lymphatic system, ends up back in the bloodstream, right? And just a general lymphatic is full of lymph fluid. It's like plasma, but lacks a lot of the plasma proteins, lacks a lot of other material that plasma has, but similar to that. Also, remember that big highlight for lymph is that lymph vessels are somewhat like veins, very vein like, and they have valves that help them return. They need valves that kind of help them get back. Okay. So kind of keep moving forward. If you look at page 345, we start talking about our lymphatic tissues. You can break up our lymphatic system into lymphatic tissues and lymphatic organs. Okay. Excuse me. So, lymphatic tissues, starting off, you think of tonsils. We technically have three different types of tonsils, but the big ones to highlight are what's known as the pharyngeal tonsils, which is known as the adenoids, right in the back towards our nasal pharynx, towards the back of our hard palate. Palatine tonsils, okay, those are the traditional tonsils we think of on either side of our throat, either side of our uvula. Those are the big ones I would highlight. Other examples of lymphatic tissues are on 346, appendix, and Peyer's patches. Peyer's patches hang out around our small intestines and large intestines and kind of keep an eye on our bacterial health of our gut and keep an eye on that for us. Then talking about lymphatic organs, you've got thymus, spleen, technically also included lymphatic organs. You would also thymus, spleen, excuse me. Also, you would include uh, bone marrow, because bone marrow is where that stuff is formed, and how our health of our bone marrow plays a huge role in the immune system. Again, lymphatic and immune system are like brother and sister, very closely related. Thymus, big thing to remember with thymus is that is where T cells mature. Thymus was a T cells mature in the thymus. Again, the thymus gland is that gland that's very large as a newborn and to infancy, and then slowly gets smaller as we get older, especially really starts to get small after puberty, is in the mediastinum, directly kind of above the heart, around the aorta. And that gland's role is really immune system focused, even though it's technically an endocrine or gland. Its goal is to be a spot for to make T cells and for T cells to mature. Thymus T cells. Okay. So I'm kind of going through this fast. Sorry, I wasn't clicking my slides. I apologize for that. Again, when we do this 20-minute lecture, it's fast paced. We're skipping through slides. We're kind of going through stuff. Okay. So there's your thymus. Again, spot for T cells to mature, hang out, get exposed to blood and pathogens. Included in our lymphatic organs, along with spleen, thymus, bone marrow, is lymph nodes. Lymph nodes are discussed on page 349, 348. So lymph nodes, again, really their big thing for lymph nodes is just they're not all this structure, but they are dotted throughout the lymphatic vessels, and they're just a spot for filtering. Okay, they're filtering out toxins or filtering out pathogens, holding it. They're big spots for our white blood cells, immune system cells to kind of mature, as well as just get trained up. Okay. So they house a bunch of lymphocytes and monocytes. That job is to kind of clean everything. And we have a few big spots that I talked about you want to know for lymph on page 348 at the body of work at the bottom. Cervical lymph nodes, axillary, cervical around the neck, axillary underneath the armpits and around the breast, and inguinal lymph nodes. Lymph nodes kind of around the groin. And page 349 talks about lymphadenopathy, swollen lymph nodes. Okay, they're large or swollen lymph nodes, usually as a relation to infection. So swollen lymph nodes, you think infection, but downstream from that, right? So if my inguinal lymph nodes are swollen, the lymph nodes around my groin, I may have an infection, some kind of issue going on downstream from that, which would be somewhere in my leg. Okay, so kind of put it that way. And that's why with like a really bad upper respiratory infection, we could have cervical swollen lymph nodes. See, and this is that picture that's on for lymphadenopathy discussion on page 349. The spleen, kind of the unforgotten organ of our system, but lymphatic organ, it's the largest, hear me there, it's the largest lymphatic organ. So the spleen is the largest lymphatic organ. Big thing for the spleen is discussed on page 350. Body of work section and a life lesson section down there. Body of work, the spleen is big in immunity. Again, lymphocytes, specific white blood cells to lymph, and macrophages hang out there, get trained up, get exposed to blood and other pathogens, destroy stuff they need to. Other big things your spleen does is it destroys old red blood cells, helps break them down. And blood storage, it stores a large amount, 20-30% of our platelets. And also it helps a little bit with producing red blood cells. If in like infancy, in the it helps produce red blood cells as we're a fetus and into infancy a bit. And then it's in reserve for producing red blood cells if we become really injured and hemorrhage a lot, lose a lot of blood, become very anemic. It can kind of kick in and create red blood cells. But its primary is immunity, being a spot to train white blood cells, specifically our lymphocytes, destruction of all red blood cells, and the other big thing is blood storage, and that's where the splenic rupture comes into play. So spleen is in a spot kind of right up under the rib cage that is usually in motor vehicle accident, steering wheel hits it or something like that, and it can get ruptured or there's a lot of bleeding involved with a splenic rupture, and it can be very scary and deadly even because it's so vascular, and it's a place that stores a lot of products that make blood and platelets and stuff. So it's very bad whenever it gets injured. So those are big things for that. Second half of chapter 17 is all immune system. Okay. So immune system, again, 3 lines of defense we talked about in the first. Really what I want to get into is this non-specific immunity. Okay. So non-specific immunity is talked about starting kind of page 352 onward. So 352 on their book talks about phagocytes, which include in that is macrophages, which are just big phagos. And remember phagocytosis, right? Cell eating. Phagocytes are just big white blood cells that roam around or hang out in certain tissue types. They're going to eat pathogens or foreign materials. And neutrophils, our first responders. Hey, neutrophils are in the phagocytes category. They're also just kind of our most numerous white blood cell first responders. They're kind of the first ones on the scene. They're going to swallow stuff up, destroy pathogens, be there to help us. Moving on, another subsection of this is that antimicrobial proteins that's discussed on 353. So antimicrobial proteins include a couple key ones is really interferons, or interferon is a type of protein we have. And they kind of help target receptors on neighboring cells, kind of let everybody know, like, hey, there's an infection here and you guys should do something about it. So interferons are kind of like the messaging system. Again, this is all under the umbrella of non-specific immunity, meaning that non-specific means that it doesn't know memory of a virus. If it sees the flu, it doesn't know what it is. It just knows it doesn't belong and it needs to do something about it, right? And the interferons are just circulating the bloodstream. And there's a picture kind of what they're doing on page 353. The other part of that is these other microbial proteins, antimicrobial. Again, antimicrobial is anti a microbe. So microbe would be a foreign pathogen, something antibiotic, excuse me, bacteria, virus, whatever. Anti that is anti those things. Complement system. The complement system is the other. So interferon, complement system, or complement proteins are proteins that our job is floating around the bloodstream to do non-specific immune functions. Complement proteins, big thing. They're usually circulating the blood inactively, and then whenever there's an immune response that gets triggered, they get activated, and their way that they destroy pathogens, they actually punch holes. Okay, they burrow holes or punch holes, and that causes fluid to shift in and out of the cell wall of a virus or a bacterium or whatever, and that's just how they destroy things. So you got interferons that kind of bind to the surface and trigger like a wave of white flag, like, hey, come here and do immune response. And then you have complement proteins or the complement system that float around the bloodstream and active. And when they get activated, they punch holes in whatever they're trying to target, like a bacteria or whatever. Also, want to mention the bottom of page 353, we have natural killer cells. This picture talked about here on this slide. Natural killer cells, again, or not again, but natural killer cells are, they get confused with killer T cells that we'll talk about later. Very similar, but natural killer cells are technically lymphocytes. They're lymphatic white blood cells. Natural killers are what they sound. They're like the assassins of our immune system. Okay. They're just like roaming the body. They're small enough to kind of roam throughout the bloodstream, and they're just seeking out pathogens, that sort of stuff, cancer cells, anything they can find out that seems unlikely or not supposed to be there. Okay. And they kind of get moved around. So they're usually breaking apart that cell and destroying it by lysis. Last couple of things are discussed on page 354 and 355 for non-specific immunity. This is no memory, no anything, just anything foreign that's not supposed to be there gets attacked. Inflammation is one of those. Inflammation is just any trauma, injury, ischemia, so no lack of oxygen causes inflammation. Inflammation's job is really to stimulate the whole body's kind of fighting something off and kind of help clean up and repair. And there's a diagram that shows that on page 354. When you think of inflammation, you think of that bottom section, a body of work, swelling, redness, heat, pain. These are all signs of inflammation. And those are related mainly to like fluid moving out of the capillaries, increased heat and movement of stuff, just trying to get an reaction to happen at a certain injury site. Okay. That's inflammation's job. Another thing with inflammation is that if you think about inflammation or infection, sometimes we think about pus or abscesses. That's discussed on 354. Really what pus is that yellowish discharge from like an infection spot. That's just buildup of a bunch of dead white blood cells, other debris, platelets, combination of inflammation, those kind of things. And they just create this thick yellowish fluid called pus. It's just a combination of all the dead material from fighting off infection. And if that pus it accumulates in a body cavity somewhere or something like that, like in a tooth, right? You get a tooth abscess. That's what an abscess is. So abscess is just a buildup of pus somewhere it shouldn't be, basically in a cavity. That's discussed on that page. And then finally, fever, pyrexia is another name for fever, pyrexia. That's why antipyretics treat fever. Okay. You'll learn about that later. But so if a person has a fever, they're said to be febrile, Febrile, febrile. So if you don't have a fever, like I don't right now, I am afebrile. So really fever is a little bit neutrophils and those other guys release something that kind of causes fever, a fever producing substance called pyrogen. I would highlight that pyrogen, pyrexia, all related to fever. Okay. So basically, fevers are beneficial, generally, especially the low-grade ones. They're trying to produce an uncomfortable environment for bacteria or pathogens like viruses, whatever, to live. And that's what a fever is doing. It's fighting off infection. That's generally a good thing, but we can also treat that. So talking about specific immunity is what I want to talk about next. Skip through this stuff, kind of going fast. So specific immunity, the big things with that is how we break up specific immunity. Specific immunity is, again, talking about there's usually memory made, and that way we can fight stuff off later on. Excuse me. So the big thing is at the top of page 356 talks about cell-mediated immunity or cellular-mediated immunity and humoral or antibody-mediated immunity. So I usually refer to them as cellular immunity or humoral, Humoral immunity, humoral immunity, okay? That chart at 356, the body of work section down there about active and passive immunity, though I guarantee those will be exam questions. So active and passive can be broken up into natural and artificial. Natural active is in the sense of your body naturally producing antibodies after you've been infected with a strain of flu or something like that. Artificial active is still active. It's an active process. Your body is making antibodies, but artificial active is like when you get it inoculated, you get it vaccinated with like a flu vaccine that is an attenuated virus. So it makes your body produce antibodies against those strains of viruses. It's like introducing it, but it's artificial, right? Because we manufactured it. The other side of that is there's a natural and artificial passive. Passive immunity is just antibodies being passed to somebody. There's no work. There's no active process of your body making antibodies. You're just given the antibodies at the time. So the big examples of that natural passive is any antibodies that pass the placenta during fetal development, or any antibodies that are passed from breast milk from mom to baby, basically. So mom to baby is passive, natural passive, and artificial passive is like, for example, a snake bite or anti-venom for something, or things like the rabies and botulism. You would get the direct, someone would inject you with antibodies to fight those processes off, like an anti-venom. You don't make antibodies against the venom, but you're given antibodies, or what treats the venom, or treats the disease process. And that's artificial because we made it. And it's also passive because your body doesn't make anything for it. So you got to know those. Page 357 is lymphocyte discussion. T and B lymphocytes. Okay, big thing is how you memorize these is that makes sense section on page 357. You've got how to differentiate T cells and B cells right there. So T cells mature in the thymus with a T, right? B cells mature in bone marrow, right? Bone marrow with a B. Okay. So T cells are usually manufactured in the thymus. Most of the time, the T cells are going to mature in the thymus. B cells, though, usually begin their life as stem cells in bone marrow, and they stay in bone marrow until they are fully mature. Unlike T cells can either be made in the thymus or made in bone marrow, but they're always going to end up in the thymus to mature, where B cells are going to be made in bone marrow and stay there to mature. So B for B cells, bone marrow. T for thymus, starts with a T for T cells, okay? And they kind of end up going all different places: spleen, tonsils, lymph nodes. But their goal, as we're going to talk about them, is how we make antibodies, and that's the part of the humoral antibody mediated immunity, okay? So antibodies, another name for them, okay? They're floating around in plasma, made from B cells. Antibodies come from B cells. Antibodies come from B cells, okay? So the antibodies discussed on page 357 as well. The other name for antibodies is called immunoglobulins. Immunoglobulins, okay? IG. And we have a bunch of different IgA, IgD, IgE, IgG, IgM, all those guys, okay? Those are interesting. You do not have to know those, but we make a bunch of different immunoglobulins, and that's what they're talking about, different types of antibodies. They're made from B cells. And again, remember antibodies from our discussion about blood and blood typing. Antibodies' job are anti something. So they look for an antigen presenting on a cell, and they go and say, hey, I know that antigen, right? And I don't like them, like our red blood cells. And it sees it as either it's checking that antigen as self or non-self. And it says, if that antigen shows non-self, I'm going to attack it. Okay. I'm going to bind to it and destroy that thing. So page 358, 359, excuse me, summarize all that. Okay. So the big summary for cellular immunity and humoral immunity, big things, big takeaways is that you've got to kind of break down these images, right? So let me get there. Sorry. So cellular immunity is this image here on page 358. I mean, the big thing is kind of having an idea what the difference is between cell immunity, cell mediated or cellular immunity, and humoral. Is again, cellular immunity is more associated with the T cells, and humoral immunity associated with B cells, because the end result is that with humoral immunity, we're producing antibodies for long-term memory, for long-term fighting stuff off, right? So T cells, there's three different kinds of T cells, and that makes them confusing. There's cytotoxic T cells, also called killer T cells, not to be confused with natural killer cells or NK cells. So cytotoxic T cells kind of float around a little bit like NK cells, but their job is to kill. Helper T cells are just supportive. And then memory T cells actually hold on to a memory of what they've been like in fighting with, okay? So they can retain a memory. So they're basically T cells are programmed to attack any cell that's just not redeemed itself. So basically, what T cells are doing, if something gets swallowed, a helper T cell, or excuse me, T cell is going to come in and go, hey, what is that? And look at the cell, and it's going to present that antigen. It's going to line up with the antigen. And then that T cell disperses and makes those three types. It makes a T cell, cytotoxic killer T cell, to go out and fight that thing it just learned about. It makes a helper T cell to go assist in humoral immunity and make antibodies. And then it makes a memory T cell to kind of hang out long life. So we switch over to humoral immunity and look at that, right? Again, focuses on stuff outside the cell, focuses on antibodies to mark an antigen for destruction by other cells. That's the goal. Antibodies slap to an antigen and they're just saying, hey, this is time for destruction, right? So, big thing there is that B cells contain thousands of receptors for specific antigens. And when they kind of catch on to that antigen, they're like, oh, what is this? Okay, right? The B cell is going to kind of engulf it. And then with the assistance of that helper T cell binding to it, they're basically going to reproduce a bunch of B cell clones that are going to differentiate. These B cell clones they make right here that I'm doing with my mouse. Okay, these are highlighted as known as plasma cells. That's outlined in the fourth step here, plasma cells. So they make a bunch of helper T cell helps them identify this pathogen after they broke it up and then make a bunch of B cells that eventually turn into plasma cells that eventually turn into what we know as antibodies that have memory, long living, and their all antibodies only are harmful to the antigen that they were created to target. Okay. But they're going to trigger things like agglutination. You're right. They're going to tell complement proteins to come in. They're going to help trigger interferons to come in and trigger the immune response. Like they can alert the whole immune system, mark a cell like an anti or a microbe of some kind and say, hey, destroy it. And they're also going to make another memory cell that floats around for long-term immunity. Okay. So B cells end goal is long-term immunity, long-term memory, antibodies to mark stuff outside a cell and destroy it. T cells are do a similar job, but they're going to make 3 separate versions of themselves that all have a specific role. Those cytotoxic T cells, helpers, and memory T cells. The other big thing, highlight from the immune system, is immune system disorders. It's talked about on page 361. And then transplantation and tissue rejection is talked about on 362. I would have a little bit general idea of that. But autoimmune diseases is talking about my body attacking myself for some reason, whatever it is. Our body is making antibodies that attack our own tissues. This is what happens to type 1 diabetics. That's an autoimmune condition attacking their B cells of their pancreas. They can't make insulin, right? So that's what immunodeficiency or autoimmune diseases. Common one that's discussed is HIV in there, but HIV is interesting too because the HIV virus actually invades and targets T cells, helper T cells, and that kind of just messes everything up. Because T cells play a role in both humoral and cellular mediated immunity, so that's why they're so bad. So we're going to skip through our questions and get on to chapter 18. And we're going to be way over time. So it's going to be more like a 30-minute lecture, but that's okay. We'll make sure you get the information, right? So chapter 18, respiratory, page 367. Basically, this is a good snapshot of our respiratory system. My focus for this is going to be anatomy. You can break up a respiratory system, into upper and lower respiratory tracts, right? So a big thing here, okay, is that you're looking at the upper respiratory tract consists of things that you think of, right? Nasal cavity, nasopharynx, oropharynx, laryngopharynx, and the larynx. Okay. So you look at that upper respiratory, it starts getting broken down on page 368. I think the big things there is that you have to think about the different subsections. So we have our nasal passages, nasal cavities. They have what's called conchi in there, to help direct airflow. And they have all their cells are mucus kind of membrane cells that line with cilia, right? Can catch dust. And then we have our pharynx is the next process, right? So we go into our pharynx that has 3 subdivides. You're going to need to know all those 3 subdivides: nasal, oropharynx, and laryngeal pharynx, all kind of subdivided from the pharynx. Larynx is, you think, voice box, okay? Larynx is where our vocal cords are housed. Also, the spot where epiglottis is that covers up our trachea here. So the larynx is kind of the opening where the larynx starts is where our epiglottis is. And the epiglottis has the ability to kind of, it's a muscular connective tissue flap that will close over the trachea and let liquids and foods go back towards the esophagus and keep them from going in our lungs. Okay. But larynx, you think epiglottis, vocal cords. Pharynx, you think about the 3 subdivides that are kind of the back of the tongue and throat and the back of the nasal cavity. Okay. Lower respiratory tract is fairly straightforward. You have the trachea on down from that. And this is discussed on page 370. So you have the trachea down from that. and you have the primary bronchi after that. Again, think of the lungs, the lower respiratory tract, as this subdivide is trachea, the two big bronchi, and all their subdivides, and then just the lungs themselves, right? So if you look at the lower respiratory tract, it looks like a tree, right? It's got that very tree. My brain just shut off. Sorry. So it looks like an upside down tree, right? And that's the thing with it. Okay. So really, the big thing is you think primary bronchi, left and right. Key thing that I would make a point here, okay, is that a really interesting fact. You can see on this image here that this right side of our lungs, our right bronchi, okay, is way more, it goes, has a way more verticality to it, okay, where the left is more horizontal. And it's also a little bit like steeper and a little bit wider. And so if someone swallows something, if they get something stuck in their airway and it makes it past the trachea, like food or other particles, if it's called silent aspiration or aspiration, that's what it's called when you swallow something in your lungs. If that food stuff makes it, it's probably gonna end up in the right side, and that's where people get right lung infections because particles or whatever made it down in here, and then they get infection from it. So it's always not always, but usually gonna end up in the right side. Okay, especially I'm talking about food and stuff like that. So next, I want to mention alveoli. So alveoli is discussed on like 371. Basically, alveoli is the name for these small sacs at the very, very tiny microscopic end. And this is where kind of the bread and butter of the lungs happens. This is where the oxygen exchange happens. You can see pulmonary capillaries from our pulmonary circulation, which is a portal system, right? Remember that? So each alveoli, you imagine like these big, they're just these sacs that are kind of filled with air that compress and expand based on our breathing. And their job is to house a bunch of capillaries on top of them. So we do that exchange of oxygen and CO2. Okay. Again, our veins are bringing CO2 back, right? And our lungs' job is to get rid of it and then bring in fresh oxygen. Okay. That's the job. Again, kind of looking at the lungs, I think it's really important that you would know asthma, and we'll have a discussion about pneumothoraxes. Those are on page 372 and 373. I do want to discuss pulmonary ventilation. It's broken down starting on page 375, but pulmonary ventilation is basically talking about, hey, okay, how we breathe, what are we doing, right? How does it work? Basically, the two subdivides of pulmonary ventilation: inspiration, I'm inspiring, I'm breathing in, and expiration, exiting, expiration, out. Normal range, respiratory range per minute for an adult is about 12 to 20 breaths. That's a good thing to know. Okay. And the other thing I really want to talk about with pulmonary ventilation is more discussed on page 378. Talks about pressure and airflow. I'm trying to get there. Sorry. And again, what's the big, one muscle that we talk about? I've talked about multiple times that without it, you don't breathe. Diaphragm, right? So diaphragm is based or not diaphragm, but pressure and airflow is based on atmospheric pressure. This is discussed on 378. So basically, what you have to kind of imagine here is that looking at this image, inspiration. So breathing in, right? The intercostal, so some of our muscles around our chest, and are going to contract, and they kind of pull the ribs up and out, and the diaphragm moves down. Okay, so inspiration, the muscles around our ribs are pushing out to expand our lungs, and the diaphragm moves down. That during inspiration. All right. And this is kind of happening because of the atmospheric pressure around us, the weight of the air outside of our body. Okay. So basically, you would think when the pressure in the lungs drops lower than the air outside, air is going to flow from an area of high pressure to the low pressure. Okay. So that's kind of inspiration. You think of the higher pressure of the air outside the body rushes into the lungs because the lungs are now lower pressure. And that's inspiration. The opposite is for expiration. When the lung pressure inside of our lungs rises above our atmospheric pressure, the pressure outside of our body, that forces the air out. And that also it's just not a muscle thing, but it's a atmospheric thing. And this is the negative ventilation is why the lungs are talked about being a negative pressure system. Okay. So basically, is what's happening here is that the pressure, if you have to imagine at the peak of like expiration, getting all my air out, now all of a sudden the air pressure in my lungs is way lower than the atmospheric pressure around them. And this kind of tells my lungs and forces my diaphragm down, ribs out, and that pushes air, rushes in, and does all this at the same time. On the other end, expiration is the opposite. Whenever my now all of a sudden, the very peak of inspiration, like my lungs are as full as they can be, right? My lungs are full of air. Now the pressure is higher inside my lungs than it is outside in atmosphere. That causes the lungs to, my ribs to kind of go back in, diaphragm to push up, and then push air out. And this is what causes breathing. So it's not just muscle contraction and the diaphragm moving that is needed and required for breathing, but it's this atmospheric pressure versus the pressure inside our lungs, okay, and this negative pressure system. So also another life lesson you got to look at is emphysema on page 379. We need to know about emphysema, and that discusses COPD, chronic obstructive pulmonary disease, okay. And then on page 381, there's a really decent chart, talks about common respiratory terms like apnea is temporary cessation of breathing, cessation, so stopping breathing. So that's what sleep apnea is. Sleep apnea is you stopping breathing while you sleep. That's a problem. You don't want to do that, right? Other than that, the other stuff is not too concerning. I would look at page 384 and discuss kind of talking about factors that affect gas exchange. We really want gas exchange to be happening, meaning that we want as much potential, right, of CO2 getting out and O2 getting in, right? I mean, that's kind of ultimately the goal. I do want to draw your attention back to page 379 when the discussion about alveolar surface tension is on 379, just above emphysema there. Alveolar surface tension, that discussion, okay? Again, that's talking about what helps. The alveoli are covered in this thin film of water liquid, and it has a little bit to do with chemical attraction and chemistry. But what forces kind of keeps the water from moving around and just let the water, the fluid from the lungs and the fluid that is in the capillaries, what kind of keeps that is surfactant. It's a type of lipoprotein that basically messes up electrical signals, and the surfactant surrounds all the inside of the alveoli, and it basically just lowers surface tension and kind of helps this electrical impulses and keeps the fluid where it's supposed to be. So if you think surfactant, okay, you think what lines the alveoli and keeps the fluid where it's supposed to be. It's based on chemistry and kind of electrical signals and stuff with molecules, but the overall arching thing is, hey, okay, this is what it makes it breathe. So that is your quote unquote 20-minute summary of chapters 17 and chapters 18, which is all unit 7. Okay. So if you have any questions after hearing this, again, please let me know. Thanks for taking your time to listen to us. And I hope you guys have a great evening, and God bless.